Good morning all. Welcome to the presentation. In this presentation we are going to discuss on the topic sterilization and disinfection. So first one is sterilization. It is a process where all living microorganisms including bacterial spores are killed, and the sterilization can be achieved by physical, chemical and physico-chemical means. So there is a chart of different methods in the sterilization. We can see in detail. First one is physical methods. Physical method include sunlight. First one is sunlight. That is the microbial activity due to presence of UV rays in sunlight. Next one is heat. Heat is most liable method. It include dry heat, moist heat. First one is dry heat. Red heat. It is red heat. Flaming, hot air oven, infrared rays, incineration are examples in the dry heat. Next one is moist heat. At the temperature, it includes at the temperature 100 degree and below 100 degrees Celsius and above 100 degrees Celsius. Next one is radiation. It includes ionizing and non-ionizing. Ionizing, which means high energy ray with high penetrating power. It includes particulate and electromagnetic. Next one is non-ionizing. It is low energy ray with poor penetrating power. Next one is filtration. It does not kill microbes, only separates them out. Examples: earthenware filters, asbestos filters, sintered glass filters, membrane filters, etc. Next one is vibration. This sonic and ultrasonic vibrations that kill some bacteria and viruses. So the physical methods are completed. Next one is chemical methods. It includes liquid and gaseous. Liquids, first one is alcohol. It is dehydrate cells, disrupt the membrane, causes the coagulation of the proteins. Example is ethyl. Next one is aldehyde. Act through alkylation of amino carboxyl or hydroxyl group which damages nucleic acid. Example formaldehyde. Next one is phenol. Disruption of membranes, precipitation of proteins and inactivation of enzymes. Example 5% phenols. Next one is halogens, oxidizing agents and causes damage by the oxidation of essential sulfatryl groups of enzymes. Example is chlorine compounds. Next is heavy metals, act by precipitation and oxidations of sulfhydryl groups and which is bacteriostatic. Example mercury chloride. Surface active agents, property of the concentrating at the interfaces between the lipid containing membrane of bacterial cell and surrounded aqueous medium. Example soaps and detergents. Dyes. Acridine dyes are the bactericidal because of their interaction with the bacterial nucleic acid. Example is aniline dyes. So next one is gaseous. So the liquid types completed. Next one is gaseous. First one is ethylene oxides, that they are alkylating agents, acts by alkylating sulfhydryl amino hydroxyl groups. Next one is beta propiolactone BPL, alkylating agents, acts through alkylations of carboxyls and hydroxyl groups. So the sterilization are completed. Next one is the disinfectants. So the definition of disinfection is that the process of killing or inactivating harmful and objectionable bacteria, cysts or other microbes by various agents such as chemicals, heat, ultraviolet rays, waves or radiations. Disinfection is usually considered a 99% kill compared to the sterilization that generally attains 100% to kill. Next one is the disinfectant's mode of action on microbes. It includes growth inhibition and lethal action. Growth inhibition means bacteriostasis or fungistasis. Lethal action, it is bactericidal or fungicidal. So first one mode of action, the first one is action on the external membranes of bacterial wall. The quaternary ammonium compounds that bound to the phospholipids and proteins by creating permeability of the cells. Next, action on microbial wall. The chemistries like amphoteric compounds easily punctuate the cell wall, and the amino acid property of these compounds enable to enter the cell wall and the cytoplasmic membrane. Next one is action on the cytoplasmic membrane. The PHMB causes the destruction by rupturing the membrane and cell permeate without lysis of the cell wall. Next one is action on energy metabolism. Exposure of the microbes to the lethal doses of hypochlorites causes a decrease in ATP production. Next one is next action on the cytoplasm and nucleus. High levels of disinfectants chemistries like parasitic acids, hydrogen peroxides are active in the degenerating the nucleus and DNA. Next action on bacterial spores. Active disinfectants can destabilize the structure in spores and mostly preferred for their eradication. And another topic is method for testing disinfectants. First one is carrier test. It is the oldest test and then here is the bacteria which is used in this culture. Two observations get. First one is growth occurs and no growth occurs. So another topic is method for testing disinfectants. First one is carrier test. It is the oldest test and the bacteria which is used in this culture is mentioned. So two results are obtained. First one is growth occurs. Next no growth occurs. The growth occurs means that less or ineffective disinfectants, and no growth occurs include more effective disinfectants, and this test can be used for the liquid disinfectants. Next one is suspension test. The sample of bacterial culture is suspended into the disinfectant solution. After the exposure, it is verified by the subculture of bacterial culture. Growth occurs, less effective disinfectants. No growth occurs, more effective disinfectants. Capacity test. This test is determine the capacity of disinfectants to retain its activity in the presence of increasing contamination load. Bacterial suspension added until disinfectant capacity to kill is exhausted. Best known capacity test is Kelsey-Sykes test. Next one is practical test. This tests are performed under the real life conditions after ensuring some time concentration relationship of the disinfectants under the quantitative suspension test. It is done to verify if selected dilution is still adequate under the conditions under which it must be used. Best known practical tests are surface disinfection test. Example for it is time kill test. Next is in-use test can be used in hospitals and laboratories to detect to the contamination of disinfectants. 1 ml of the sample disinfectants added to the 9 ml of diluent. 10 drops of diluted sample are placed on each 2 nutrient agar plates. Incubated at 37 degrees Celsius for 3 days. The other incubated at room temperature for 7 days. 5 or more colonies on each plate indicate the contamination. So this is all about the different modes of disinfectants tests. Thank you.